Right. MK-104 is a small molecule drug that has been in clinical use in Japan since the 1980s as an intravenous anticoagulant therapy. It's soluble in aqueous solutions and can be delivered simply to the airways as a nebulized aerosol to treat patients. Cystic fibrosis is a multi-organ genetic disease where chronic lung disease is the major cause of morbidity and mortality. In Europe and the United States, it is the commonest of the genetic rare diseases, as defined by orphan drug legislation on both continents. The genetic lesion causes defective transport of salt, sodium chloride, across epithelia. This includes the cells that line the airways. And the genetic defect specifically causes transport of chloride ions to be impaired. But as a physiological consequence of this, in the airways, absorption of sodium ions is excessive. Excessive absorption of sodium causes failure to clear secretions. And mucus secretions are part of a sophisticated refuse disposal system that keeps the airways clean and healthy. Inhaled debris, including microorganisms, is trapped by secretions and is normally removed by coordinated beating of cilia that are present on specialized surface cells over the majority of the airways. Excessive absorption of sodium in CF airways causes disengagement of the interaction between mucus and cilia. And this disengagement causes this essential refuse disposal system to underperform in cystic fibrosis. There are two CF therapies in widespread current use that partially downregulate sodium absorption in the airways. They are inhaled hypertonic saline and bronchitol, which is inhaled mannitol. These therapies have achieved modest efficacy to improve mucociliary clearance and also modest clinical benefits in phase 3 clinical trials. MK-104 also acts to reduce excessive sodium absorption, but by a very different mechanism. And what I want to show you today is that it achieves a superior preclinical pharmacology profile compared to these existing treatments, better efficacy, and much longer duration of response. Our key assay to enable us to do this is to repeatedly measure over hours ciliary transport speed ex vivo on pig tracheas. In this graph here, we can see that treatment of control tracheas with balanced salt solution resulted in a gradual decline in transport speed over a 6-hour period. However, treatment of tracheas with hypertonic saline stimulated ciliary transport speed to peak at about 1.5 hours and then decline back to baseline at 4 hours. Treatment with mannitol produced an almost identical response. By contrast, treatment with MK-104 resulted in a response that was slow onset, but it was of greater efficacy and considerably longer in duration. Ciliary transport speed did not increase above baseline until about 1 hour after treatment, and then it increased steadily to peak at 4 hours, and thereafter it started to decline. But please note that the response was still well above baseline at 6 hours after treatment. MK-104 produced a similar response profile in tracheas from transgenic CFTR knockout piglets. And here the response was repeatable after a second treatment at 15 hours. We've also obtained evidence of long-duration enhanced ciliary transport after in vivo inhalation of MK-104 in terminally anesthetized pigs. We can therefore conclude that across a range of assays, MK-104 has a superior preclinical profile compared to existing treatments. This suggests that MK-104 is a treatment candidate with promising potential to deliver superior clinical benefits to all CF patients, irrespective of genotype. And we look forward to being able to move forward to evaluate clinically this promising repurposed drug. Thank you.